So obviously, we know that this is a really society of aging. Everybody wants to live longer and healthier. Obviously, there's a way that we can do nowadays. Um, One important question is always being asked, from the outsider, is how long we can live. If you look at the carefully of the human life expectancy, it looks like over the last 50 years, life expectancy has been steadily increased, but somehow at some certain point, it's no longer being continued, but rather than decline or stable. So what that means, people have been challenged this because they believe that we can live for 150 years or 300 years by modifying some of the functional inside of bodies or stem cell replacement or organ replacement. So hopefully, in the future there will be something coming out to achieve this goal. And another interesting observation, over the years, is that women always outlive to men. We have also been looking into this for many years. Obviously, there's some clue, but it's not actually fully understood. Obviously, the X chromosome has some role to play in the longevity or life expectancy. But so far, little advance has been made, and I think this is a very exciting area that in the future we can tackle these questions. Many people believe that aging is genetics. I think this is true to a certain extent. So this is an example why actually genetics is important in determining how long you can live. Take a look of this ladies, the most long-lived human beings in the world, who actually have very bad lifestyle, heavy thinker, heavy smoker, but she managed to live over 110 years. So that really tells us, genetic is important. If you have actually good genetics, you may be able to live much longer. Of course, evidence has appears with the study of the Joyce family, centenarian family from Professor Yu-Hsin Su's work. Obviously, genetics is important in this family, particularly Joyce family here. This family really has some mutations in important genes that determine the aging and lifespan, which is insulin is factor-like receptor 1. So the mutations in the receptor 1 will basically reduce the metabolism rate, so that actually increase their health span and lifespan dramatically. This whole family basically can live over 100 years. So it's very rare. Further evidence that's showing that human longevities or human aging process is very much determined by genetics. It's the study from various human premature aging with certain mutations that will cause maintenance of genome stability or DNA repair fidelities, where lead to accelerated aging process. There are many such examples in humans. One of the most well-known examples is the Hashimoto-Gilford syndrome, which results from heterozygote mutations in the nucleolaminar protein called lamin A. Patients suffer from various, we call segmental progeria phenotype as early as 1 to 2 years old after birth. if you look at the nuclear shape from the patients, very much like nuclear shape that we observe in the elderly person. So it really represents a significant portion of the normal aging process. Therefore, has been using as a good example or good model system for aging research. Now, even though we believe that genetics play an essential role in determining how long we can live, but obviously there's something else can also affect the aging process and how long we can live. So the important evidence comes from the animals. So we all know that the bees, the queen bees basically can live for 6 years. Well, the drones, worker bees, can only survive for 6 weeks. Now, genetically, they are exactly the same, but the only difference is the food, right? So likely that food will result in the difference in the epigenetics so that controls the aging process, so they can live much longer. So indeed, actually, from our own study and many other studies, actually, if you modulate the epigenetic, including the methylation, histone modifications, etc., then you can actually change the fate of the cells in terms of cellular senescence or DNA repair. For example, in this case, we use the sodium butyrate to treat the mice with suffer from Hutchinson-Gilford-Crutcher syndrome. In this case, we also use the XAM24 knockout mice, which actually also results in unprocessed mutant lamin A. And the consequence is actually the DNA diamond repair is actually rescued, and also the cellular senescence has been suppressed, and you have a much longer lifespan. You have about 25 to 30% increase in the progeria mice. So that really tells us genetics is important, but epigenetic can also play a significant role in determining our lifespan expectations. So over the last 20 some years, we all know that there is well-studied mechanism behind the aging process, and we have now identified 12 different hallmark of aging. If actually you categorize this hallmark of aging, you can be categorized to primary hallmark, antagonistic, and integrative hallmarks. So if you look at the primary cause or changes in aging, they all related with genomic maintenance and the protein stasis. So they are the central of the aging process, but not necessarily that you can target only on the primary hallmark and achieve life expectancy expansions. But you can also target the other hallmark of aging, and particularly when you target inflammations, and you target the mitochondrial dysfunctions, etc. You can also actually achieve a significant extension of the lifespan. So one of the hallmark of aging is the cellular senescence, which is actually one of the core hallmark of aging. And when we think about the cellular senescence, we always think the bedside, right? But actually, the nature has developed senescence as a protective mechanism to prevent the cancer development. So that's why actually, we are getting old because we need to prevent the cancer development. So we try to avoid getting cancer at the expense of getting old. So, it's good and bad. You don't want to get cancer, but you have to get old. So the proof of concept that really, by eliminating senescent cells can protect our, health span and extend our lifespan come from this study, which basically use animal model, premature aging models, try to eliminate the P16 positive cells that were actually resulting significant extension of lifespan in the progeria mice. So that's actually the classical proof of concept that by eliminating senescent cells can extend lifespan. Why actually senescence is so bad? Because when you have senescent cells, you also always have these inflammations. So the inflammation results in the accumulation or recruitment of the inflammatory cytokine from the immune cells. And what happens to the local fibroblasts? They start to proliferate and they start to secrete all kinds of extracellular matrix, collagens, etc. That actually really make the tissue become dysfunctional. Okay, so it depends on the severity of the inflammation or proliferation of the fibroblasts. It will become either has a condition or a disease. If you are able to suppress inflammation by removing all the dead senescent cells, your tissue become actually regenerate again. So they can self-repair and regenerate. So the functional tissue become restored. So there are actually lots of evidence, studies that shows already indeed, aging process, there's increased inflammations. For example, this study from the 13-6 null mice has shown that basically if you loss of 13-6, which results in accelerated aging process in animals, it's because the lose repression of the Y1 retrotransposon, so which is actually start to become much active and in lots of transcription going on. Eventually, it causes actually activation of the inflammations. So you can either introduce the 13-6 back again, or you can use inflammatory inhibitors. So you can achieve basically the life extension and suppress the various aging phenotypes in the 13-6 non-cow mice. So there's actually a lot of senolytic has been developed to target the senescent cells, try to suppress the inflammation and extend the lifespan. So we also worked on one of the component, which is called the cannabidiol, which is CBD, as a senescence killing substances. So you can see basically if you treat the senescent cells with CBD, they basically can remove all the senescent cells, largely from the culture cells and also from the tissues. So if you actually feed the animals with CBDs, for example, either in the progeria mice or in the worm, C. elegans, they can actually significantly increase lifespan, especially average lifespan. If you take the cells out and do the colon inflammations, CBD can also increase the colon inflammations to protect the stem cell becoming senescent. So here you can see actually some of the tissues, especially subcutaneous fat, has coming back after the CBD treatment in the progeria mice, which usually has completely loss of the subcutaneous fat. And also the cellular senescence has been suppressed. So that really tells us CBD can actually serve as senolytics. Actually, one of the main functions for CBD previously being reported is as a suppression of the inflammation. So now we know at least one of the mechanisms for CDD to suppress inflammation is through the elimination of senescent cells. So if you actually look at the transcriptome, you can see basically over almost 80% of the genes, are differentially transcribed in the premature aging cells now can be restored back to normal by the treatment of CDD. And we also figure out that there are actually several pathways important. If you take away, by knocking out all these key pathway genes, then the CBD will no longer treat the removing senescent cells, and they no longer behave like the senolytic. So that really tells CBD can actually work through their receptors and talk several pathways that eliminate or kill the senescent cells. Over the last few years, people has also tried, another way to kill the senescent cells apart from senolytics. Now, senolytic has side effect in general because most of senolytic developed so far are actually from existing drugs, especially chemotherapy drugs. So even though they are, preferentially kill the senescent cells, they actually also exhibit cytotoxicity to the normal cells. That's why actually if you apply the senolytic to animals, they always have side effect. So another strategy now being developed is to try to use immune functions to eliminate senescent cells specifically without targeting the normal cells or young cells. So to achieve this, we need to identify the sort of xenoantigens specifically expressed or highly expressed in the senescent cells, lower expression in the younger cells. So if you be able to identify those senescent cells antigens, then you will be able to develop different strategy targeting the senescent cells by either CAR-T, CAR-NK, or antibody, or even the vaccines. So by targeting these senescent cells, with or without tissue specificity, you may be able to target different age-associated diseases, so like hypertension, diabetes, obesity, etc. So based on this concept, there are actually several papers recently published showing some of the surface protein in the senescent cells are actually able to serve as xenoantigens. For example, the Japanese group published a paper recently, 2 years ago, which target immune cell surface marker called the GPNB, and they actually show there's a significant improvement of the metabolic diseases in the animals feeding with high-fat diet. Also, another group recently targeting UPAR, which is usually is a cancer target. They use this target to eliminate senescent cells, which can also achieve a significant improvement of the aging phenotype in the animals. Now we also did screening of the xenoantigens using cells, animals model system, and also human tissues. We identified over 100 different xenoantigens as our potential target, and then we went through all these identified, validated the expressions of the xenoantigens by comparing the gene expression in the elderly or senescent cells with young cells, tissues, and we actually identified several important targets that has not been reported before in the aging research. You can see actually one of the targets that's shown here is highly expressed in the tissues of adipose tissues in the old animals, but not in the young animals. And then we actually go through, first round screening to test whether it can be used as a target for intervention of the aging process. So the animal model now we're using is actually the high-fat diet animal models. So using the metabolic diseases as a model system to see whether some of these targets can be used to intervene the metabolic diseases. Indeed, at least 2 or 3 of these targets showing significant beneficial effect when we use peptide vaccine to treat the animals. So basically, you can see compare with the untreated high-fat diet fed animals. The animals with injection of one of the targets has actually significant reduction of the body weight over a few months. And when you look at the fat tissues, after sacrifice of the mice, you can see they actually contain less senescent cells by looking at the staining of the tissues of the beta gall stainings. And you can also see they actually achieve a significant reduction of the glucose, blood glucose levels. And when you look at the safeties of these peptide vaccines, they actually show significant safeties without damaging the kidneys or liver. Actually even improve the liver functions by reducing some of the parameters. So you can also see they reduce the senescent cell, number of the senescent cells. And you can see we use the reporter mice, which shows adipose tissues and senescent cells in the abdomens. You can see by the treating of the animal with the vaccines, you can achieve a significant reduction of the fat. So they actually are very good for obesity treatment in the animals at least. So you can also see that the fat livers has also been significantly restored or rescued in the vaccine-treated animals.